Class 11 Biology Chapter 6 Anatomy of Flowering Plants Part 1 You can very easily see the structural similarities and variation in the external morphology of the larger living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cells as a basic unit. Cells are organized into tissues and in turn tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures are also show adaptations to diverse environments. 6.1 The tissues. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissues. Tissues are classified into two main groups, namely meristematic tissue and permanent tissue, based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissue. Growth in plants is largely restricted to a specialized region of active cell division called meristems. Plants have different kinds of meristems. The meristem which occur at the tips of fruits and shoots produce primary tissue called apical meristems. Root apical meristem occupies the tip of a root while the shoot apical meristem occupies the distant-most region of the stem axis. During the formation of leaves and elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axil of leaves and are capable of forming a branch or a flower. The meristem which occurs between mature tissue is known as intercalary meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristem and intercalary meristems are primary meristem because they appear early in life in a plant and contribute to the formation of primary plant body. The meristem that occurs in the mature region of roots and shoots of many plants, particularly those that produce woody axis and appear later than primary meristem, is called the secondary or lateral meristem. They are cylindrical meristems. Fascicular vascular cambium and interfascicular cambium and cork cambium are examples of lateral meristems. These are responsible for producing the secondary tissues. Following division of cells in both primary as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissues. During the formation of the primary plant body, specific region of the apical meristem produce dermal tissue, ground tissue and vascular tissue. 6.1.2 Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and function are called simple tissues. Permanent tissue having many different types of cells are called complex tissues. 6.1.2.1 Simple tissues. A simple tissue is made of only one type of cells. The various simple tissue in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma forms the major component within the organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma performs various functions like photosynthesis, storage, secretion. The collenchyma occurs in layers below the epidermis in dicotyledonous plants. It is found either as homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to deposition of cellulose, hemicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant such as young stem and petiole of a leaf. Sclerenchyma consists of long, narrow cells with thick and lignified cell walls having a few or numerous spits. They are usually dead and without protoplast. On the basis of variation in form, structure, origin and development, sclerenchyma may be either fibers or sclerites. The fibers are thick-walled, elongated and pointed cells, generally occurring in groups in various parts of the plant. The sclerites are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities lumen. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear and sapota, seed coats of legumes and leaves of a tea. Sclerenchyma provides mechanical support to organs. 6.1.2.2 Complex tissues. The complex tissues are made of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissue in plants. Xylem functions as a conducting tissue for water and minerals from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of four different kinds of elements, namely tracheids, vessels, xylem fibers, and xylem parenchyma. Gymnosperms lack vessel in thick and in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layers of the cell wall have thickening which vary in form. In flowering plants, tracheids and vessel are the main water transporting elements. Vessel is a long cylindrical tube-like structure made of many cells called vessel members, each with lignified walls and a large central cavity. Vessel is a long cylindrical tube-like structure made up of many cells called vessel members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protein. Vessel members are interconnected. The presence of vessel is a characteristic feature of angiosperms. Xylem fibers have highly thickened walls and obliterated central lumens. These may be either septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made up of cellulose. They store food minerals in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cell. Primary xylem is of two types, protoxylem and metaxylem. The first form primary xylem elements are called protoxylem and the later formed primary xylem is called metaxylem. In stems, the protoxylem lies towards the center pith and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endark. In roots, the protoxylem lies towards periphery and metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transports food material, usually from leaves to other parts of the plant. Phloem in angiosperm is composed of sieve tube elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperms have albuminous cell and sieve cells. They lack sieve tubes and companion cells. Sieve tube elements are also long, tube-like structures arranged longitudinally and associated with the companion cells. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The function of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The sieve tube elements and companion cells are connected by pit fills present between their pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in the secondary phloem. These are much elongated, unbranched and have pointed, needle-like apices. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax and hemp are used commercially. The first formed primary phloem consists of a narrow sieve tubes and is referred to as protophlloem, and the later formed phloem has bigger sieve tubes and is referred to as metaphloem. 6.2 The tissue system. We were discussing types of tissue based on the types of cells present. Let us now consider how tissue vary depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue system. These are epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue systems. 6.2.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which forms a continuous layer. Epidermis is usually single-layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stroma is composed of two bean-shaped cells known as guard cells, which enclose stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer walls of guard cells away from the stomatal pore are thin and the inner walls towards the stomatal pore are highly thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are together called stomatal apparatus. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongation of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. Trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.2.2 The ground tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissue such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary rays. In the primary stems and roots, in leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called the mesophyll. 6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stem, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues, and hence are called open vascular bundles. In the monocotyledonous, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues and they are referred to as closed, when xylem and phloem within a vascular bundle are arranged in an alternate manner on different radii, the arrangement is called radial, such as in roots. In conjoined type of vascular bundles, the xylem and phloem are situated at the same radius of the vascular bundle. Such vascular bundles are of common in stems and leaves. The conjoined vascular bundle usually have the phloem located on the outer side of the xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stem and leaves, it is convenient to study the transverse section of the mature zones of these organs. 6.3.1 Dicotyledonous root. The transverse section of the sunflower root, the internal tissue organization is as follows. The outermost layer is epidermis. Many of the epidermal cells protrude in the form of unicellular root hairs. The cortex consists of several thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water, impermeable, waxy material subornin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjugative tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis such as pericycle, vascular bundles and pith constitute the stele. 6.3.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundle and pith. As compared to the dicot root which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. 6.3.3 Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem show that epidermis is the outermost protective layer of the stem, covered with a thin layer of a cuticle. It may bear trichomes and a few stomata. The cells are arranged in a multiple layers between epidermis, pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of few layers of parenchymatous cells just below the epidermis, which provide the mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded thin-walled parenchymatous cell with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains and the layer is also referred to as the starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is a characteristic of dicot stem. Each vascular bundle is conjoined, open, and with endark protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. The monocot stem has sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. 6.3.5 Dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface and the lower surface of the leaf, has conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of the spongy parenchyma. It has 2 types of cell, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. 6.3.6 Isobilateral monocotyledonous plants. Leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic difference. In an isobilateral leaf, the stomata are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles except in main veins, as seen in vertical section of the leaves. 6.4 Secondary growth. The growth of the roots and stems in length with the help of apical meristem is called the primary growth. Apart from the primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called secondary growth. The tissues involved in the secondary growth are the two lateral meristems, vascular cambium and cork cambium. 6.4.1 Vascular cambium. The meristematic layer that is responsible for cutting of vascular tissues, xylem and phloem, is called vascular cambium. In the young stem, it is present in patches as a single layer between the xylem and phloem. Later it forms a complete ring. 6.4.1.1 Formation of a cambial ring. In dicot stems, the cells of the cambium present between primary xylem and primary phloem is the intrafascicular cambium. The cells of the medullary rays adjoining this intrafascicular cambium become meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. 6.4.1.2 Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells both towards the inner and outer sides. The cells cut towards the pith mature into secondary xylem, and the cells cut off towards the periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. The primary and secondary phloem get gradually crushed due to the continued formation and accumulation of secondary xylem. The primary xylem, however, remains less or more intact in or around the center. At some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial direction. These are secondary medullary rays. 6.4.1.3 Spring wood and autumn wood. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform through the year. In the spring season, Cambium is very active and produces a large number of xylary elements having vessels with wider cavities. The wood formed during this season is called the spring wood or early wood. In winter, the cambium is less active and forms fewer xylary elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has lower density, whereas the autumn wood is darker and has a higher density. Two kinds of woods that appear as alternate concentric rings constitute an annual ring. Annual ring seen in a cut stem gives an estimate of the age of the tree. 6.4.1.4 Heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the central or innermost layer of the stem. These substances make it hard, durable, and resistant to the attacks of microorganisms and insects. This region comprises dead elements with highly lignified walls and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as sapwood. It is involved in the conduction of water and minerals from root to leaf. 6.4.2 Cork cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermal layers get broken and need to be replaced to provide new protective cell layers. Hence, sooner or later, another meristematic tissue called cork cambium or phellogen develops, usually in the cortex region. Phellogen is a couple of layers thick. It is made of narrow, thin-walled and nearly rectangular cells. Phellogen cuts off cells on both sides. The outer cells differentiate into cork or phellem, while the inner cells differentiate into secondary cortex or phellodome. The cork is impervious to water due to suberin deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phellogen, phellem, and phellodome are collectively known as periderm. Due to activity of the cork cambium, pressure builds up on the remaining layers peripheral to phellogen, and ultimately these layers die and slough off. Bark is a non-technical term that refers to all tissue exterior to the vascular cambium, therefore including secondary phloem. Bark refers to a number of tissue types with periderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. At certain regions, the phellogen cuts off closely arranged side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped opening called lenticles. Lenticles permit the exchange of gases between the outer atmosphere and the internal tissue of the system. These occur mostly in woody trees. 6.4.3 Secondary growth in roots. In a dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem, bundles a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular. Further events are similar to those already described above for a dicotyledonous stem. Secondary growth also occurs in stems and roots of gymnosperm. However, secondary growth does not occur in monocotyledonous. Summary. Anatomically, a plant is made of different kinds of tissues. The plant tissues are broadly classified into meristematic and permanent. Assimilation of food and in storage, transportation of water, minerals and photosynthesis, and mechanical support are the main function of tissues. There are three types of tissue system: epidermal, ground and vascular. The epidermal tissue system are made of epidermal cells, stomata and the epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle and pith. The vascular tissue system is formed by the xylem and phloem. On the basis of the presence of cambium location of xylem and phloem, the vascular bundles are of different types. The vascular bundle form the conducting tissue and translocate water, minerals and food material. Monocotyledonous and dicotyledonous plants show marked variation in their internal structure. They differ in type, number and location of vascular bundles. The secondary growth occurs in most of the dicotyledonous roots and stems, and it increases the girth of the organs by the activity of the vascular cambium and the cork cambium. The wood is actually a secondary xylem. There are different types of woods on the basis of their composition and time of production.